So we are moving on to our next unit. Our next overall unit is Unit 6, Gene Expression and Regulation. This is our next overall unit. But first, before we even are going to talk about this stuff, we need to revisit something. It's quite important. DNA and RNA structure, what these molecules look like. This is the first topic in this unit. It's going to be very important to understanding how gene expression and regulation work. So I imagine that's why they put this in this unit. So before we talk about that, we'll talk a little bit about the history of DNA and what we knew, kind of approximately when we knew it. Of course, in the Darwin in Darwin's time and Mendel in these late 1800s or mid to late 1800s, 1860s, approximately. Not much was known, but along came a guy. This was a Swiss scientist named Friedrich Miescher. He kind of was the first person to isolate and document isolating DNA, which is a pretty common process. You could do this with household items now. But he was the first person to isolate it and recognize that there was this kind of phosphorus and nitrogen rich material that he was getting out of these out of some blood cells when he took some blood samples. Kind of the first time he actually noticed it. Now, did he know what it did at this time? No, didn't know much about it. But this was around the era of Darwin and Mendel. This is late 1850s, I believe, when he was kind of working on this stuff and isolating it. And then early 1900s, this German scientist, Albert Kossel, he was the first one to isolate the actual nucleotides, ATCG. So we went from knowing that, hey, there's a lot of phosphorus and nitrogen, whatever this is, to, hey, we have these, actually, five if you count uracil over here, this stepchild of this RNA nucleotide. But he went ahead and isolated these actual bases and was able to recognize that there's these different bases in DNA. Still didn't know much about the structure of DNA other than it had a lot of phosphorus, a lot of nitrogen, and it had these unique bases as part of their structure. Along came these guys. You've probably heard of them before, maybe not. Watson and Crick, credited with the discovery of the structure of DNA. Everything about they kind of figured out that it's a double helix structure, and kind of how this structure was arranged. They use some X-ray crystallography photographs taken by, you've probably heard of this woman as well, Rosalind Franklin and Maurice Wilkins were two that were working together, taking these X-ray crystallography images, and this image kind of unlocked for these guys the structure of DNA. Of course, there's a lot of controversy surrounding these guys, especially if you look at James Watson. He's known to be kind of persona non grata amongst the scientific community. He's has some kind of views which are looked down upon, some racist and sexist views, which you might want to be interested to look up this guy and see what he's like. But yes, it was kind of a combination of all these people and many others that led to the discovery of the actual helical structure of DNA. Before this Photo 51 came along and was analyzed, these guys kind of thought that structure of DNA, they kind of knew it had some sort of backbone structure, but they thought that the backbone, the phosphorus, was actually on the inside and the bases were on the outside of DNA. And this picture kind of cleared that up for them and showed them truly what this looks like. It also gave them some measurements. They were able to measure based on the distance between these markers here, how far apart each base was. This unit here is called an angstrom, and it's incredibly small, if you can imagine. It's 1/10 billionth of a meter, I believe. 3.4 angstroms between each base, and as you can see, there's 34 angstroms in one full kind of turn of this DNA, right? It's kind of the amplitude of this DNA, if you will. So if you do the math there, you'll notice that that's around 10 bases. It's actually a little bit over 10 bases. The diameter of DNA end to end is about 20 angstroms. 10 for the radius right there, as you'll notice. So these were kind of some interesting measurements to come out of the structure of DNA. Before we move on to the other stuff, I do want to point out some differences in DNA between prokaryotic cells and eukaryotic cells. If you look over here on the left-hand side, I want you guys to look at these two images. I want you to tell me which of these do you think is a prokaryotic cell. Go ahead and pause if you need to. All right, you should have said that this one on the left here is a prokaryotic cell or a bacterium. This one on the right over here is a eukaryote. So eukaryotes have their DNA not only contained in a nucleus, but if you'll notice, over on the eukaryotic side, we have a linear chromosome style DNA. And the DNA inside of bacteria is circular. It is circular. Bacteria also has unique sets of DNA, these smaller sets of DNA, circular DNA floating around in here that are called plasmids. Plasmids. And this gives them a lot of their variety because they're able to trade these plasmids amongst each other. So two very different systems we have over here. The rest of this stuff we are going to talk about in this unit. All of this stuff down here, you will understand by the time we get to the end of this unit. This is part of transcription and translation. For now, don't worry about that because we've got to talk about DNA replication first. Okay, so moving on to structure, these are some things you should remember. DNA contains a phosphate group, right? A nitrogenous base and a sugar. Now, these two pieces here, the phosphate and the sugar, sometimes called the sugar phosphate backbone, form the backbone of DNA, as you might guess. Of course, if you're looking at a DNA molecule here, this is my crudely drawn one, but they would be the kind of these lines here. They would be represented by these lines forming the backbone of the DNA. Of course, on the inside of this, these little lines drawn towards the inside are bonded nitrogenous bases. And this is what actually gives us our code. This specific base over here is adenine, of which there are 4 bases, if you'll remember. So let's talk a little bit about these bases. Again, what I'm talking about next is this green section of the molecule right here. We have something called, and we talked about this earlier in the year, and I'm just going to put a dividing line here, pyrimidines and purines. If you'll notice, I'm going to start with purines down here. You need to know that these are 2-ring structures. Pyrimidines are 1-ring structure. I remember this by thinking the longer word actually has the smaller structure, and the shorter word has the larger structure. So purines have 2, pyrimidines have 1 ring. Their basic structure is shown below. Of course, you'll see where some of these specific purines and pyrimidines vary is these functional groups that come off of the main ring structures themselves. So let's go ahead and take a look at these. Pyrimidines, cytosine, uracil, which is found only in RNA, and thymine, which is found in DNA. Again, these two kind of swap out for each other in their corresponding biomolecule. In RNA, you're going to find uracil instead of thymine. Now, the way I remember which ones are pyrimidines is I remember these letters C, U, and T. And to me, the pyramids look like they were cut, out of something, right? They look like they were sliced or cut, C-U-T, pyrimidine. That's how I remember them. The purines, a lot of people remember these by purines, pure silver. And silver, of course, is Ag on the periodic table. So A and G, these are the purines. These are the purines. Again, they have a double ring structure, and the place they vary is these functional groups that come off in purple and pink over here, and the same goes for these pyrimidines over here. So you should know pyrimidines, cytosine, uracil, and thymine. These are pyrimidines. Purines, adenine, and guanine. So please know those. Know which one's a 2-ring structure, which one's a 1-ring structure, and know which ones belong to which group. Now, do you need to know the specific, differences between these? In other words, this one has, an NH2 coming off. This has just an oxygen over here. No, you don't need to know that stuff. But it's interesting to note that they do vary by these functional groups that come off and actually form these hydrogen bonds. So as far as structure is concerned, that's going to be it for this video. In the next video, we'll start talking about DNA replication. Okay, thank you.